Thank you all for hanging out till the end here, and to the organizers for inviting me to talk. Today I'm going to talk about our precise prophage mapping and some of the applications that we can do because of the database that we have. So I'll talk a bit about the software tools that we have and some of the tools we've developed for phage engineering applications. And then I'll talk about our phage factory, which we view as a bacterial-agnostic engineering and application development platform, and then about a class of satellites we could find because of this database. So I don't think this audience needs this introduction, but it's to remind me to tell you that our software actually finds genomic islands, which are this larger class of which prophages are a subset of. So we look for everything that has an integrase, and then we call prophages based on their genetic content. So we really wanted to go at this question on how can we find a phage for every application in a cultured way. So kind of the traditional way is looking for phages that will lyse your host of interest. And this is not always host adapted. And to date, there's about 17,500 unique cultured phages in GenBank. And we kind of took the question, if we look in bacterial genomes, these are nets that can catch phages. And typically these phages are more host adapted. We're choosing them from close relatives or from our species of interest. So how does our software work? So our software is actually looking at this integration event. This is doing that weird thing again where it's shifting things. And we are searching precisely for the @L and @R sites combined with the integrase protein. So we have two complementary software. The first is Islander, which searches for this motif that's found in bacterial genomes where you have a full tRNA gene, a tyrosine integrase, and a displaced tRNA fragment. And what that allows you to do is really map that @L and @R site within the bacterial genome. Then utilizing this information, we actually built Tiger and then followed it up with Tiger version 2. And what this is doing is it's taking a seed integrase HMM. So this can now look for tyrosine and serine integrases. And then we do a ping pong blast search where we take queries from the left and the right of our integrase of interest, and we blast it to a reference genome. So it's a comparative genomics tool. We then expect if there's a genomic island to see this broken hit, we prepare back adjacent queries and blast them back. And then this way we can really precisely map those @L and @Rs for our integrases of interest. So we had done a small scale study, which was published a couple years ago, of about 2,000 genomes. And then we really wanted to scale this up to the entire tree of life. So to date, we've treated all 48,000 GTDV species that are comprised of over 350,000 genomes. We found just under 1 million genomic islands, so 2.8 genomic islands per genome, and we found about 382,000 prophages, which averages to be about 1.1 prophage per genome. And then we wanted to further investigate this. So we broke this down into bacterial taxonomy by phyla. And we can see that this is very different across the host taxonomy. So just to give everybody kind of an overview of this, prophages are all prophages that are found. We have these things that are tandem. So half of it looks like a prophage, half of it looks like an ice. We cannot tease them apart yet. We have integrative conjugative elements, and then we have a class of things that we currently cannot classify based on gene content alone. And you can see in groups where we have lots of bacteria and well-represented bacteriophages, you get lots of prophages per genome. And then in classes that we know less about, we have less prophages per genome, although none of these groups have 0, which is really encouraging to use this approach to think about all the application spaces that we can attack. So doing all of that work took a couple years. Part of this is due to that comparative genomics method in our database. So we wanted to design smaller databases to speed this comparative genomics approach up. So we tested this on Salmonella genomes, and we designed 3 databases, the full set of 58,000 Salmonella genomes, a database with 200 members, and a database with 500 members. And we did this by maximizing diversity through MASH scores, and we left 10% of our database to be out of the GTDV species tested, so that we weren't missing some genomic island that was present in all the members of that species. And thankfully, the speed up was quite a bit faster. So the original took about 700 hours, where the 2 database speed ups took 1 to 3 hours total. And when we lose genomic islands, we actually lose many islands that are very small in size. So gray is always our 58,000 database, the light blue is our 500, and our dark blue is our 200. So we typically lose things that are under about 8 kilobases when we do this speed up, and we don't appear to lose a ton of things in that phage satellite peak or our traditional 40 to 50K phage peak. What we also noticed is we're actually not losing a lot of phages. So we break our phage categories down pretty far. We have a category 1, which is full length, about 25 kb or larger phages. They have to have structural proteins. They must have other phage identifiable proteins. We then have our phage 2 category, and they can have one of these classes or be smaller than 25 kb. We've recently been categorizing the innoviruses thanks to work done by Simon. We look for the ZOT protein, PFAM, as well as coat proteins. And then we break our ICEs down and our others. And we'll note that most of these genomic islands lost are actually in the other categories, so they can't really be classified. And further, when we're looking at these, they actually contain mostly non-canonical or truncated integrases. So we really do think these are a loss of non-high quality prophages and genomic islands. And in doing this database speed up, what this allowed us to do and work with Gary Trubel was to make Tiger version 2 that could search across scaffolds, and it can expand this precise genomic island detection into metagenomes. So we really needed a way to do this. And when we did this, we thought about our original question. So A is kind of the model that we were already doing. We could search for genomic islands if they were complete, and we could find them in tandem. We then added a module that now can go through the circular origin. So if you have part of your genomic island on one end of the molecule and part on the other, when you make the linear molecule and NCBI, we can now find that. And then we started looking at fragmented genomes where you could have a few different parameters here. You could have the left end and the right end on 2 different scaffolds with a missing middle, and we found that quite a bit. You can have the left end and right end on 2 different scaffolds with the full complete genomic island there, or you can have kind of the original case. And when we did this and we looked at what categories each of these genomic islands fell in, we can see that in all cases, about 40% of the genomic islands we're searching for are classified as phages. We get slightly higher bias in intact and the circular origin towards that phage 1 or those more complete phages. And that's kind of what led us to that missing middle hypothesis, is that when we're looking at this cross contig or this final mode here, we're seeing a lot more of this phage 2, suggesting there's something missing there. And then surprising to us, we found about 2 times more genomic islands when we use the split mode as opposed to the intact mode. So we go from about 3.4 GIs per genome in isolates to 5.8. And then the other thing that was really interesting to us was that MAGs tended to have lower genomic island counts compared to the isolated genomes in that same GTDB species. And this could be due to low sequence quality. It could be due to really, MAGs having less genomic islands. So that's still under investigation. So now that we have this database of 1 million genomic islands, what can we actually do with it? And what does that allow us to ask? So we can really start understanding horizontal gene transfer and the host range of these various prophages by asking what other hosts they're in and where they're at. We can start looking at crosstalk between prophages, mobile elements, and looking at the prophage genome mosaicism. We thankfully now have the mapped at sites for 1 million integrases, so this allows us to really explore integrase biology more. And we're now getting this diversity of prophages across the tree of life that's been currently well mapped in mycobacteriophages through that isolated population. And then we can start asking a lot of the questions that people have posed here. Are some of these bacterial species don't contain prophages because we have annotation problems? We don't really know what they look like in those species, or are they truly lacking from those species? Is this a biological mechanism? And then we can really start digging into mechanisms that regulate horizontal gene transfer in all these various species. So utilizing this prophage database, we actually built the Phage Factory, which is our bacteria-agnostic phage discovery and engineering platform. And the premise of this is that we can identify prophage-rich close relatives of our target. So in the first work I'm going to talk about today, we used Pseudomonas aeruginosa PA01 as our target, and we chose 2 very close relatives shown here in blue because they had high numbers of prophages. We then want to validate these phages. We don't want to take things that are decaying or maybe not active within their host or can't excise from their host. And then we can use these phages as they are in their wild type form, or we can engineer them through a variety of engineering methods and then reboot them into productive viruses. So I want to point out that prophage yields are typically higher than this 1.1 average for many of the pathogens that I'm showing here, which was encouraging to us because we really have a rich database then to harvest prophages for therapeutic applications. And because we have the precise endpoints mapped, we can actually identify these active prophages by PCR. So I'm showing the 7 prophages we ended up working with in this study. And this is the circular junction PCR. So after that excision and circularization, and you can see we get positive PCR products both in the filtrates and in the soaked plaques for all 7 of these. However, we've also developed software tools that we can do detection through deep sequencing. So after induction, we deep sequence all of our strains, and we look for this set of recombinant reads corresponding to the ATP and the ATB sites of the prophage of interest. And what this really reveals is the different induction behaviors between all of these prophage. You can see different biologies, you can see different rates of induction. But what I want to point out for at least the phages that we ended up working with is ATP is always in blue and ATP is always in red. Then we see this pretty consistent trend where the ATB and ATP are relatively close, suggesting there's some excision happening up to 1 hour. And then we get these large peaks of ATP, which suggests not only is excision happening, but we can actually monitor replication in real time. So we then took these prophages, and because we wanted to use them in a therapeutic application, we actually deleted the integrase genes, so they were no longer capable of integration. And then we did some common tests for killing ability. So we first tested them in liquid culture. We did all of the phages on their own, and then 2 phage cocktails. And compared to phage buffer alone, they all had a killing effect on Pseudomonas aeruginosa. We also tested all of these in Galleria melanella waxworm phage therapy trials. So the graph is flipped here because we're looking at survival. But again, compared to PA01 with buffer only, we see that all of the phages, as well as the phage cocktails, have an effect on survival, increased effect on survival in these worms. So this was really good, but we designed all of our Gibson assemblies by hand, which takes a lot of time. You have to go back sometimes. So we ended up developing a software called Big DNA, which actually does this large Gibson assembly design for you through backtracking and recursion algorithms. So if you have a phage as shown here in A and you want to break it up into multiple pieces, doing this by hand, you would do part A, then part B, then part B2, and then part B3. And if you get to the end and your primers don't work, you have to go back and start again. So we developed an algorithm where we can now take a set of primers that will go to the very left end of your genome, and you can then look for the next primer, your reverse primer here. And if that does not work, we can go back and we can do recursion until we get the full data set. And this doesn't work with just wild type phages. We can actually use this to delete regions of genomes. We can use it to do gene additions, and we can use it to do deletion addition together. So we've then moved away from more of the therapeutic bacteria to energy applications. All of the organisms up here, we are working on finding prophages for and engineering these prophages for various application spaces related to energy. I'm going to show a snippet of data on the Burkholderia cepacia complex, largely because these are found in soil consortia. They're still relevant to cystic fibrosis patients, but they're also extremely hard to transform. So that allows us some additional application space. So we obtained 16 different Burkholderia cepacia strains. All the ones in blue are what we had in our collection. And this is a tree of the Burkholderia cepacia family. We discovered 123 genomic islands, and 39 of these were prophages. By those metrics, 30 were these active full-length phages, and we could show definitively that 20 are induced by mitomycin C, 6 were under decaying state, and 3 were filamentous. However, when we looked at the diversity of these phages, we noticed that they're actually quite diverse. These 39 phages only form 6 clusters with 21 singletons, and this is after dropping the average nucleotide identity down to 90%, spanning half of the genome. And then we asked, do these look like phages in the environment? And many of these clusters do not, suggesting there's a lot of diversity as well within these prophage groups. We then started isolating the prophages. So I'm showing 2 strains here, the canonical Cephalocepacia K56 on the left, and then Cepacia 1829 here. And we can find 3 different phages on these as shown here. These are from very close relatives of the Cepacia strain. And this is from actually a multivarians, which if we go back, is way up here, and K56 is down here. So there's a lot of host range breadth that we can start exploring. And now we can start exploring with that large phage data set. What are some of the mechanisms regulating that host phage action? And then finally, with our prophage database, we can start discovering new things. So when we had our original database, we identified this very small genomic island, 11 cap E, which to us was very weird because it integrated into a capsid E protein of another phage in the genome. And then as we started investigating this further, we realized that it looked a lot like the PKs discovered in various organisms. However, there was no sequence similarity except for this very small region. So we wanted to explore this further to say, okay, well, what is this? Is this a new class? Is this just a misnomer? Is this misassemble? And what we realized is it actually is integrated into the capsid protein of its probable helper phage, which looks very similar to this Peaky's helper phage. It also has a lot of similarity to phage lambda. And then we took that integrase and we went through our entire database to ask, can we find other members of this group in other or the same gene in various lamboid type integrated phages? And we found 491 of these helper embedded satellite phage-induced chromosomal islands in different Enterobacteriales species shown here in 14 different cognate sites in lambda, suggesting that this is a real mechanism that's happening. And we have some data that shows that the entire motif or the whole genomic island 60 kb thing induces together, and then the satellite induces out of that, leaving behind the full phage genome. So today I showed you that we have prophages and we can mine them for all the known bacterial species, that our Tiger-2 revealed that MAGs contain less genomic islands than isolates. We can engineer prophages to treat Burkholderia, both in vitro and in vivo. These Burkholderia prophages have very large host strains, appears, and Hess peakies can be found in a number of lambda cognates and are widespread in Enterobacteriales. I have to thank the team that I have at Sandia and elsewhere, especially Kelly Williams, who collaborated very heavily on many of these projects, and then our funding from DOE and Sandia National Laboratories. I'm not sure if there is time for questions, but I'm happy to take some.
Yeah, no, we made up time, so
Okay.
we have time for questions.
That was really cool. Thank you. The observation that you reported in Burkholderia, where the prophages did I misinterpret this? They didn't look like the phages that had been isolated from the environment?
Not in the Burkholderia groups that we were looking at. And again, we're working to take that out. We did these 16 strains with those 39 phages initially, and we're working to kind of take that out through the whole set. So I think we have almost 3,000 Burkholderia sapacea genomes, and I don't even know, probably 3,000 or 4,000 prophages. So we're going to do those. We're actually in the process of doing those
I guess
right now.
the question is that generalizable? I mean, when you find lots of new prophages, does that mean that what we thought we knew of phages from the ones that we've been able to isolate is horribly incomplete, or is that just something specific that's happening in Burkholderia?
I would say probably both. So in some groups, like, I mean, I showed there's tons of lamboid phages in Enterobacteriales species. So we know how to isolate that. We understand what that looks like. But I think as you get into some of these more novel and wild bacterial groups, what you're seeing is they don't look like what we're culturing out.
I see.
And we are currently working on a lot of that. Like, can we take these prophages and find a host for them, or find ways to actually culture them in the lab?
Very
So
cool. Thank you.
remains to be seen. Hopefully, yes and no.
Okay, great. Any more questions? If not, we can move on to the roundtable. All right, thank you so much.